everyone for the seminar with Professor Anvesha Sarkar. So this is the seminar on the topic tackling oral tribology problems using 3D biomimetic tongue-like surface, and we have with us the awesome professor, Professor Anvesha Sarkar, and I would like to give a few introduction, a few points about our research and our career. So, Professor Sarkar, research lies at the crossroads of experimental colloidal science and human health, with the focus on understanding the mechanics and governing principles of multiphasic food colloidal structure, physiology, interaction to solve grand health challenges. And she also does a lot of experiments and interactions at various length scales with respect to nano to human scale. The experimental approaches span from use of mechanical measurements with respect to tribology, rheology, nanotribology, using atomic force microscopy. Uh, so Professor Sarkar also, she's a principal investigator of the ERC European Research Council starting grant funded projects called LubeSat from 2017 to 22, and to discover about the oral lubrication at macro to nanoscale. And also, she is a editorial board member of journals like Food Hydrocolloids, the Journal of Texture Studies, etc. And she's also been recognized by the Women Achievement of Role of Honor in 2018 at the University of Leeds. So, and also she has the prestigious Royal Society of Chemistry a medal with respect to Food Group Junior Medal in 2019, and previously, she was also awarded the Elsevier Young Scientist Award, Nestle Discovery Award, etc. Professor Sarkar, we are so happy that you are here with us. Over to you.
Okay. Thank you very much. I will share my screen. Thank you, Manoj, Adir, and Xavier for doing this. It's a very exciting thing that you guys do. And so hopefully you can all see my screen. Okay, yes. So let's start. So first of all, good afternoon, good morning, good evening, wherever you are around the globe. So thank you for joining. So what I will do today is I will try to talk to you about oral tribology, but specifically I will focus on the soft surfaces in the mouth, not so much on teeth, jaws, and so on, mostly on tongue surfaces and how basically we can design surfaces. Those are pretty exciting soft surfaces to understand more about mouthfeel. So what happens in the mouth when we eat food or drink a beverage and so on. So before starting about the oral tribology, I first wanted to understand, because I remember the first time when I started this work in Leeds, when I said oral tribology, and people were working on cars and on, gears and engine oils, and they were surprised, oral and tribology, how does that all fit in? So I always started with this very nice animation where you can see this yellow material, which is basically a food and saliva mixture, which is like a bolus, what you eat. And what is the interesting point of this animation is that this material, if you can see the yellow material, is actually touching a lot of surfaces. So, it's not in isolation. It's always touching these surfaces, which are soft, like your palate, your tongue. It is compressed, squeezed before getting swallowed. And talking from a very food perspective, we always think food in isolation. We do a lot of bulk rheology to understand how these materials flow, how they deform. But we never think about in which mouth this material is in. Like if I eat a food, it might taste very different from when you eat it, because the surfaces are very different. The salivary properties are very different. So it was very exciting at that point to understand that how surface can play a role, what you perceive. So Again, if you think of a food, it's actually broken down across the length scale. So if you're thinking of what you're eating in the beginning, a block of cheese, it can break down into, emulsion scale, as you can see, from maybe submicron to hundreds of micron. But once you start swallowing, eating, then there are remnants, which can even be in nanometric length scale. And that can create a perception. For example, when you drink wine, you have this small nanometric scale complexes, which gives you a gritty or astringent mouthfeel. So all of this are very important because this is not only a bulk interaction, but also surface interaction. So then the most important thing is look at the surfaces, what you have in the mouth. And again, I'm talking about soft surfaces. And here you can see a very nice diagram of a tongue surface, and you have these features. And what we did is we took the mask of a tongue surface and looked at under a 3D optical microscope. And here you can see there are these bumps, and these bumps are fungiform papillae, which contain your taste buds. But there are numerous of these filiform papillae, the cone-shaped or the, more like spikes. They do not have any taste buds. So it's not about taste perception, it's about friction, what they do. And basically, you can see here the scale. We are talking here about hundreds of micron material sitting in our tongue. Now, we don't perceive it so much because it's soft, it's hydrophilic. But almost think of your tongue like a soft sandpaper, which people will not tell you, but it's pretty rough material. Now, if you think of how food has evolved, we always talk about this area where you have a huge kind of film thickness in between your tongue and palate, and bulk rheology prevails. You understand how things deform. Whereas very limited work has been done on this area where there is a very thin film, where the film is not only dictated by the film properties, but also how the film interact with all these surfaces, and particularly the soft surfaces. Now, it's very important to understand that does it at all matter in terms of real oral space? And I found this work by Loga et al. In Cambridge very interesting, where they actually modeled tongue filiform papillae, the papillae which we were talking about, the crown shaped, and they modeled it with a single cylinder, and they calculated the bending modulus, and they found that this flipping of the filiform papillae is actually in the order of 10 milliseconds, which actually fits in very well when you eat a food. Because when you eat a food, let's say a honey or a yogurt, which is 100 times thicker than water, you will have this time scale, which is 50 milliseconds. So essentially, it's a slow process. It's a very quasi-static process. So you can really follow this process, what happens in the mouth. So this is just to give you an impression that oral tribology definitely plays an important role what happens in the mouth, although way more focus has been given on rheology, rightly so, but we need to also think about this thin film context, particularly when we want to talk about perception and stuff like that. So what I thought, I don't know the audience, I thought I will give you a brief introduction of the history of oral tribology. And the entire community knows that if you think of a father of oral tribology, we can think of Joseph Kokeny, who is still a professor in Purdue. And this study was done, you can see, in 1977, so quite a long time ago, actually. And what Professor Kokeny did this amazing piece of study where he took multiple food products, like 1, 2, 3, 4. These are all food products, from smoothies to juices to milk to cheese, and he did psychophysical test and friction measurement. And what we found, that there is really an inverse relationship between smoothness and friction force. And this was a very, very nicely fitted relationship. So if you think of your tongue and palate as counter surfaces, basically you have a friction force which is directly proportional to the normal force, and mu is the friction coefficient. And what Occhini did was found this amazing relationship that smoothness is actually inversely proportional to this mu, which is friction coefficient. And slipperiness is actually much more complicated, and it has a relationship not only with friction coefficient, but also with viscosity and film thickness and the velocity of the material. So this almost changed the paradigm, changed the behavior of how we used to understand food in the mouth. However, it was 1977, and then it was not followed up that much. There were some work done in industry, but not so much going on. People in Unilever did a lot of work on using those to understand friction properties of tea, coffee, and so on, but they were not published so much. So and I was also working at Nestle, so I could not publish either of the work which I did. However, when I came to Leeds, and this was, I thought, one of the very simple yet very informative piece of work which I did with Professor Mike Bryant and Professor Ardian Morina in mechanical engineering in Leeds, where the question was very simple: that if I take a yogurt which contains 0% fat, and another yogurt which contains 4% fat, can I distinguish them? And provided they're isoviscous, so they have extremely similar shear thinning behavior, very similar viscosity at orally relevant shear rate, but can I distinguish them with tribology? So what we did first the rheology measurement using a typical controlled stress rheometer. And what you can see here is that the full fat and the low fat yogurt absolutely matches the profile. So they have very similar profiles. So there was no difference. So food industries has done a great job in absolutely matching rheology of material. So we thought, let's test this material with food, with human beings. And we did a discriminative test with 63 participants, and statistically they could easily identify. So these were blind tests, and they could easily identify that, hey, this is low fat and this is high fat. And they said that the low fat was very, very watery, non-thickened, whereas the high fat was, much more lubricious, creamy, and so on. But bear in mind, they were isoviscous. So there was no difference that could be explained by the rheological feature. Then we did a tribology. And here you can see a order of magnitude difference. So red is the low fat or fat-free, and the black is the full fat. So order of magnitude difference in boundary lubrication between full fat and a high fat. And why is that? Pretty simple. Even in the MTM tribometer, you can see oil droplets floating at the top, in particularly the black one, the full fat one. So it was very clear that you can basically understand the importance of tribology in identifying differences between products. So it can be an amazing tool to do screening. So basically that's the point, and there has been follow-up, a lot of work on that with a lot of different materials like, yogurt, custard, cheese, etc., etc. So then I thought, let's look a bit about what work has been done in oral tribology soft space in recent timelines in the last 10 years. And why it has been done? The reason is again very simple. We wanted to have correlation with what happens in the mouth in terms of sensory perception. Food development is a pretty long process. And what you do in industry is you basically test with consumer. Do you like it? There are different kinds of sensory tests. Can you minimize that? And the only way to minimize it, can you have mechanical test to understand correlation and screen the material before it goes to human test. And what we identify with a lot of our study that smoothness, creaminess are inversely proportional to friction, whereas astringency is directly. So then we had a deep look at the timeline, that how tribology has evolved. And if you remember, I told you about 1977 to 2017, this 40 years gap. Not, I would say there has been a lot of work, but not in that systematic level. But in the last 10 years, there has been tremendous amount of work on fluid gels from Birmingham, on particles from Wageningen, Jason Stokes' group in Australia on polysaccharides, our group on protein microgel, emulsion microgel, hydrogels, and so on. And we can basically say there are a couple of areas where oral tribology has a lot of things it can bring into, and that is designing healthier foods. So basically, we can use tribology as an amazing tool to create healthier food, detecting food adulteration. So sometimes you don't need those very complicated chromatographic technique to understand adulteration. Sometimes addition of just the material can raise the friction and you can pinpoint it with a tribology test. Tailoring food for vulnerable population. We are doing a lot of work in this space, particularly designing food for older adults. And finally, achieving food sustainability, which is a point which I will be talking to you about plant proteins, because here the main problem is about plant proteins are very, very astringent. And I will tell you how we have kind of addressed the problem to a certain extent. Of course, you know, you are a group of tribologists, so you know scale matters in tribology. Of course, we are talking about tongue scale, but tongue is not a one surface, you know, it has so many papillae. So we look at ball on disc tribometer, considering it's a smooth surface, but we also look at AFM. Why we look at that? Because then we can identify things at single papillae level. So not only the full tongue, but the single papillae, and we can understand those interactions. And how AFM works, it's like a braille, so it reads it by touching it, and you can get force distance curves. And we, in Leeds, what we do with physics is we develop soft colloidal probes. So we don't work on classic AFM tips, we work on soft colloidal probe and, you know, generally try to define the deformability, which is in line with the deformability of a real human tongue. So this is the full tongue scale, and this is a single papillae scale. And of course, the laws are different. For example, in the tongue scale, it's independent of the contact area. Whereas when you think of the papillae scale, their adhesive interaction prevails and you have JKR model. And that's why the contact area becomes extremely important. And we see that this also reflects. So why we all need to know this? Because we see that that varies. For example, the same protein varies differently in a full tongue scale versus a single papillae scale. So understanding multi-scale lubrication is extremely important, which is, I will say, almost at the very, very early stage, at least in food science. So what I will do with this background, I will first give you a kind of a short snapshot of what we did in Leeds to create a tongue surface, which is almost like a real human tongue, both at macro and nanoscale, which means both at the tongue scale and the single papillae scale. And then I will give you one case study where I have used this, to understand the frictional property of plant proteins and how I have solved the problem. Because as I mentioned, plant proteins are very delubricating, so they have very high friction. So how can I use these tools which I developed to solve the problem, first to quantify and then to solve the problem? So let's start with the first one, which is about designing a biomimetic tongue surface across landscapes. So this work was done by Dr. Efren Andablore, who was a postdoctoral fellow working with me. And what we started off by saying, what is our closest proxy? The paradigm is that we always work with PDMS, which is polydimethylsiloxane. And those who have worked in it, it's a pretty stiff polymer. And of course, in biological system, people say, no, it is pretty soft. It's softer than steel. It's softer than glass. Of course it is. But if you think of biology, it's far away. It's orders of magnitude stiffer than a tongue surface. So what we did, we took the closest proxy, which is a pig's tongue, because we, of course, can't ask human being to donate their tongue for this work. So we took pig's tongue and we measured their deformability. So this is a true stress strain diagram. And what you can see is, irrespective of wherever you take the analysis, they essentially are in the order of 2 to 4 kilopascal. So they are very, very, very soft. But bear in mind one thing, this softness should be taken with a pinch of salt, because this is a dead pig's tongue, which is being extracted from after postmortem. But in real human being or in real animal, when you're eating, tongue is a muscular organ. So it can actually increase in terms of the pressure and so on. So essentially, you're talking about roughly 2 to 20 kilopascal or 30 kilopascal range, whereas your PDMS is in the order of megapascal. So that's where we are starting from. So we said, okay, let's work on a new polymer, not PDMS, which is softer. And this was the new polymer which we worked on, which is EcoFlex, and some surfactant is added, and I will tell you why later. So what you can see here, this is your PDMS, which is a classic material. You're talking here about 500s of kilopascal, whereas in EcoFlex, you are getting much lower, because you're using a much, much softer polymer in terms of your true stress. So this is the first bit of the puzzle, which is the softness. What does it mean in tongue scale? You have converted the contact pressure of particularly the filiform and fungiform papillae to 33 and 9.8 kilopascal as compared to 343 kilopascal. So you're talking order of magnitude lower softness, which is closer to a real human tongue. Then the next question was, how can we make a surface which is rough? Now, if you look at literature, what people have done, they have 3D printed some dimples of 100 micron, stacked in a grid and printed it. But again, the question was, is it a real tongue? What is a real tongue? So here what we did, we went to human beings, we went to School of Dentistry, and they helped us to collect masks of real human tongue. So I was one of the participants, and there were also many others. And we took a hydrophilic mold and a hydrophobic mold and collected tongue mask. And this is the negative, so you can see holes. And if you turn into positive, you will see the dimples coming. So these are all fungiform papillae. So when we converted and when we used a 3D optical image, what we identified is, this is how a fungiform papillae looks like, surrounded by numerous filiform papillae. And these are the filiform papillae. So we now have an average size of the filiform, average size of the fungiform, height, diameter, how they are located, how far they are from each other. But now, how to create an average surface? Because it's very hard. It's not a, arithmetic mean that we create the distances and put it all together. It will not work. So then again, we went back to biology and we asked the question how biological features are generally arranged, like neurons for rats, for example. And we always saw that these surfaces have features which are always in random order. They are never in grid. So if you look at literature, all the studies done were always in grid. You put the cylinders in a grid and you print it. But this is so far away from what happens in biology. So what we did is we created a Poisson-based randomization. And we said this is a kind of a hypothesis that if we create a Poisson-based randomization, the chances are that it will replicate more the biology. And how to prove that? So we had a theory or a hypothesis that the distribution which is closest to creating mechanosensation is the right distribution. So what does that mean? So if you take a single food particle and if it hits one papillae, that means there is a mechanoreception. What is the chance to increase this hitting? Because if it hits, there is mechanoreception. So with that, we created this model. And what we saw is that we did some computational work and we saw if you do a grid, the chances of mechanoreception is 50%, 56%. But if you do a Poisson point process, you have the distribution which is in the order of 90% of chances of a particle in linear line hitting the papillae, which is actually similar to a real human tongue mask. So we are pretty confident that this kind of distribution works, and not only works, it's actually congruent to what happens in mechanoreception. So it's resembling to what happens in real human tongue. So with that, we created our first biomimetic tongue surface. So here is what the mold looks like. And then we replica molded it. And this is how the beautiful biomimetic tongue look like. And you can see the surface with the fungiform papillae. And here is all the filiform papillae. They are soft, they are rough. And now we'll tell you more about the wettability. So we again went back to the pig's tongue, and we identified that actually the wettability was in the order of the static contact angle was in the order of 70 degrees Celsius, 70 degrees. So it's not really hydrophilic, but it's also not hydrophobic. If you look at PDMS again, you have a very, very high hydrophobicity. However, with the new polymer, EcoFlex, what we did is we added the surfactant. In the beginning also, I said I will get back to you on that. So we added the surfactant to make the contact angle around similar to a real human tongue. So what we have done, we have created a soft surface. We have made it rough with the right kind of size, shape, and distribution of papillae. And the third, we have made it wettable, similar to a real human tongue or a pig's tongue. So this is how a 3D printed tongue looks like. And this is a real tongue mask. Bear in mind, there is this curvature. We did not want to have this curvature in our system because we wanted to have a proper contact. So now, the most important thing, okay, you have done a tongue which looks like a tongue, which does all the features like a tongue, but does it behave like a tongue? Because that's the most important thing. So what we did a tribology measurement, and we use xanthan gum, which is a polysaccharide. And what you can see here, this is a friction curve of a smooth PDMS of xanthan. And this is a friction curve of our real tongue mask. And this is our biomimetic tongue surface. So which was very, very promising. Whereas we did the same thing also with whey protein, which is a protein. And here again, we can see that this is a behavior of a real tongue mask, and here is a biomimetic. And the difference is largely because of the shape difference and electrohydrodynamic regime. And what was interesting is that after we have done before and after, there was not much deformation. So they were all quite good. So you can use the surface if you want to. So we're pretty excited by this work, and we thought this was an exciting piece of work done in our lab. And somehow the press was also interested to hear more about it. And there was a lot of media interest and so on. But for me, the most exciting one was Sci-Fi Ware, who said human tongue, doppelgangers, 3D printed by scientists is not scary at all, which was quite exciting. So we made it. And one of the questions which everybody was asking is, hey, that's great, but what do you do with that? This is exciting. So we said, okay, we will show you what it can do, which normal PDMS surfaces can't. But bear this in mind for a while, and I will quickly show you what we did at the nanoscale. So at the nanoscale, again, we did not do a real tongue, but as I mentioned, we were doing a papillae. And this work was done by Ivan, who was also our ERC-funded postdoctoral fellow. And we did a massive screening of the literature to identify what work has been done on soft probes. And what we identified that this is the area where we are talking about around 100 to 150 kilopascal, where there's very little work in literature on colloidal probes. So they're all in very, very hard colloidal probes. So we use the same technology, but here what we did is we actually made PDMS with different kinds of cross-linking and base ratios, and we can make PDMS surfaces and the colloidal probes also at so low stiffness, so 150 kilopascal to 2 megapascal. And they were not different in their roughness, so which was pretty exciting. When we did it, what we identified was, of course, depending upon the kind of deformability, you have very different deformation. So for example, if you have a 150 kilopascal probe, it had a very, very large deformation because it was soft. Whereas if you have a 2 megapascal or 1 megapascal slope, you have a much, much smaller deformation. And that matters because that matters in perception. And when we fitted it into a GTE model, what we identified it was actually falling into the JKR regime. So basically, and there was protein. So basically, adhesion mattered a lot in these kind of systems. Why it is important? I will quickly show you in an example that both these measurements are important, the tongue scale and the single papillae scale, because the kind of behavior that happens with proteins in the mouth are very localized sometimes, and I will show you why. So now, using these tools, what we do is we measure protein tribology. That's the main point, what I'm trying to show you. So we all know there is a huge emphasis towards transition towards plant proteins. Why? Because, food sector has done something quite dramatic that it actually contributes to a significant proportion of greenhouse gas emission. And what we have seen in literature is actually the amount of contribution of this greenhouse gas emission from animal proteins are almost twice or thrice as plant proteins. So obviously, if we move more towards plant proteins, we can have a huge impact in terms of sustainable food design. However, plant proteins suffer from a huge problem of very limited solubility, quaternary structure, they tend to aggregate, and they have a lot of astringency issues. So this is my team, and we tested almost all the products from the supermarket made up of plant-based proteins, and we tested them, and few of them are pretty awful actually. So they were dry, very, very astringent. So we were very curious, what is this astringency? And if you know the term astringency, dryness, it actually is very commonly used in wines, tea, and coffee. So I thought I will quickly show you what is astringency. So here, I don't know whether this will work. No, it's not working. Unfortunately, the video does not work, but anyway. The point is, what happens is you take an astringent molecule, it interacts with your salivary proteins, it forms these complexes and precipitates transmembrane salivary mucins, which are lubricating. Essentially, what it results is in a very, very dry sensation. So you're precipitating your lubricious salivary layer, and finally, you're getting a very dry sensation. And it's trigeminal. So in the sense, it is not a taste, but a tactile sensation. Now, the most important thing to remember here is we are not talking about small molecules. We are talking about plant proteins, which are pretty massive molecules. They not only interact with saliva, but they also interact with other plant proteins. So Ben is one of my PhD students, and what we did is we looked at tribology of alternative proteins. We used different kinds of plant proteins, and also we used some insect protein just for fun. And what we can see here, so this is a friction curve. The blue one is an animal protein, which is whey protein, which has the lowest friction. And you can see here, potato protein, pea protein, all have pretty high friction. So what we did, we wanted to understand why is it happening like that? And we did some QCMD, which is quartz crystal microbalance with dissipation measurement, to understand the film formation. And what we observed is that pea protein has a very, very high amount of adsorbed mass when it adsorbs, and it forms essentially a very interesting cross-link structure because of jamming of these particles due to aggregation. So that's the reason why it was giving so high friction. Now, that's the reason why we always do things at macro scale, at nanoscale, because when we did at nanoscale, it was exactly opposite. Pea protein had the lowest friction. Why is that? Because this film formation at a single papillae level was good enough to prevent the plowing, whereas it was not good enough at the full tongue scale. So that's the reason why I always say that these are two different things, and we cannot like really correlate, because at nanoscale, it all depends not only of surface affinity, but also the level of hydration that this layer forms. And apparently, pea protein forms a hydrated layer, which other proteins don't.
No worries, take your time, please.
What we did then was we tried to understand the sensorial property of plant proteins. And what we realized was that when you look at very high concentration, irrespective of plant protein, they have a lot of different behavior. However, when you go to astringency, they have some sort of similarity. So they indeed have a very similar kind of behavior. They are all astringent, independent of the kind of protein. We also did some brain imaging to understand what happens when you take an astringent molecule like a like a tea or a wine versus pea protein. And this is how it works. We can see here that if you take the oxygenated and deoxygenated blood, we can measure that during this behavior. And what we see is that, unlike water, pea proteins behaves like tannin, with our right side of the brain getting innervated. So it's not just a perception. It also happens in the brain. I'm extremely sorry. Now we looked also at the cellular level reason of astringency. And what we identified, that again, unlike whey protein, which is the animal protein, pea protein behaves like tannin with significant amount of binding to the cell lines. So you can see here, the tannic acid and astringent pea behaves almost similarly, with a lot of material bound to saliva. So then we had an idea how we can convert plant proteins into microgels, because microgels are blobs of water which are connected to the pea protein. And the whole idea is how can we convert plant proteins, which are very, very hydrophobic, into hydrophilic material. And basically,
Shall we take a couple of minutes break? And
Yeah, I think it's a good idea, and we can, yeah, we can also invite people to ask questions in the chat. So I think we forgot to let people know that they can write in the chat, and later on, maybe after the presentation, if we have time, we can go through the questions and see, yeah, if it's possible.
Sorry about that. That's all right.
Not at all.
No problem.
So what we can see here is, yes, we can take a plant protein and convert it into a hydrated mass. So that's the whole point. So here you can see how we can make microgels. So you actually use the plant protein and their inherent cysteine chemistry and heat treat them to form these gels. So essentially they contain mostly water and very, very little protein. And then you can make these gels and shear them up using blenders to form these microgels. And you can change their volume fraction, deformability, and so on. So what we observed is that our hypothesis was simple, that if you convert the plant protein into a microgel, you can actually increase the lubricity. So here what we can see in the rheology data, that the rheology of plant proteins changes dramatically if you make it into a microgel. It increases quite massively. So you have order of magnitude increase if you increase the volume fraction of the microgels. Now we did the tribology, so this is the AFM, so you can make them, we made it very small particles, but you can make them whichever size you want, depending on the pressure of the homogenizer you're using. And you can also make them different sizes, shapes. We were interested in making them spherical, but some of them essentially were a bit more oval shaped or elliptical shaped. And this is the most important data. What we observed is that this is a buffer, this is the native protein, and when you make a microgel, the gray one, it behaves just like a oil in water emulsion. But bear in mind, the microgel does not contain any oil. So this was pretty striking. So now we took the microgel and put it in a 3D biomimetic tongue-like surface, and we saw exactly the same behavior, that the microgel was behaving like an oil and water emulsion without adding a single drop of oil. That means you have converted an astringent molecule into a fully creamy material without adding a single drop of oil, which was very, very exciting. So it indeed improves the lubrication performance and emulating an oil and water emulsion. We also did some theoretical modeling using logistic equation. And what we see here is that the P protein, plant protein behaves like buffer. So it fits the behavior of the buffer, whereas the microgel behaves like the oil and water emulsion. So this was, again, quite a lot appreciated by the community, and they said you will make the plant-based meat more like juicy and stuff by single ingredient water. But I think there are two stories to it. One is it goes to the protein, makes it hydrophilic. But most importantly, we did some load experiments, and what we identified that these microgels were actually soft material which were squishable, but they were not destroyed. So they were keeping the viscosity constant, which is very exciting. And that's one of the reasons why you were getting such low friction behavior, which was pretty exciting. So you are creating a material which releases some amount of water, creates a hydrated layer, but completely does not destroy by shear. So that's the beauty of creating microgels. So to summarize, I will say the most important thing, which you all know in a community, is tribology is a systems property. And it's very important to get the surface chemistry, topography, and the contact pressure right, particularly for biology, because biology is very different from a typical hard surfaces. We think that microgelation offers a very unique technique and can be applied to a wide range of things, from food to oral therapies, and we are working on that. It's also important to build some mechanistic hypothesis before going for, just comparing tribology sensory relationships, because sometimes we get a lot of empirical relationship that does not mean much. And finally, saliva plays a very, very important role in food oral processing, which is often neglected. So it might be important to do all this work with tribology in presence of saliva. And I'm sure you have seen the work of Sia's talk previously on science of melting chocolate. So that's another thing to think about. The tribology can open up a lot of understanding about lubrication science, what happens in the mouth for food, which is already known food, and bring new understanding. And we are only at the very, very early stages. With that, I would like to really thank the collaborators. Particularly, I would like to mention Professor Mike Bryant from Mechanical Engineering and Dr. Simon Cornell from Physics. And they have been absolutely outstanding in building those collaborations, my team, and all the funding bodies. And thank you very much. And sorry for the inconvenience caused due to the cough. Thank you.
Thanks to you, really. Thanks for fighting it through.
Thank you. Yeah.
Good. We have some questions, so either go for it, please.
Yeah. So we get some questions in the chat, and I think it's good if we can maybe address them. John, you can unmute yourself and ask the question. Actually, he has two questions, so you can unmute yourself.
Can you hear me?
Yes, we can hear you.
Oh, yeah, thank you for the nice talk. I was curious, does EcoFlex have, after it's prepared, does it have uncross-linked polymer or solvent in the matrix? The reason I ask is because we were looking at PDMS as a biosimilar, and you can prepare it down to low kilopascal moduli, but it ends up full of uncross-linked polymer and solvent, and I've been a little bit nervous that it would, come out into the sample.
No, it doesn't. We have worked, as I mentioned to you, the nanoscale work we did with PDMS. The issue of using PDMS, which I find, is that particularly when you try to make it hydrophilic. So you can do plasma treatment to make it hydrophilic. The issue is that at a macro scale, it is very transient. So it changes, that's one. And it's very hard to control the wettability. At nanoscale, we tried it, and it actually forms a film. So it's actually, if you do the experiments of indentation, it's pretty exciting because you have a lot of data on that if you look at the film, if you indent until the film, it's very, very thick. It's very hard. The deformability is pretty low. And if you indent down, then suddenly you are in that 150 kilopascal regime. So it's pretty exciting. So working with hydrophilized PDMS is a big, big challenge. And that's why we moved to EcoFlex, and we never saw any cross-linked polymer. Yeah.
Okay, excellent. I guess I'll ask the other question too. So do you think, so you pair colloidal AFM with macro tribology. Do you think you could get similar insights by pairing colloidal AFM with shear rheology instead? Or is it just fundamentally different in that respect?
I think, means to a certain, of course, we use shear rheology, but where I struggle is the thin film properties, to be honest with you. And we have seen most of the sensory perception is actually from those after films, which are in the almost molecule level thickness. versus what happens in the shear rheology, where you're talking about at least, hundreds of micron scale most of the time. So I think it's a scale dependency rather than anything else. And we have seen the relationship with sensory happens more towards the boundary to mix lubrication regime, which works brilliantly with macroscale tribology.
Thanks so much, Dr. Sarkar.
Thank you, John. Thank you, Professor. And we have another question. You can unmute yourself and ask the question.
Hi, can you hear me?
Yes.
Hi, thank you for a very interesting talk. I'm a fourth year PhD student at NC State, and I work in rheology primarily. I'm fascinated by the work that you're doing, and I wanted to know, since you've had extensive industrial experience as well, what are the differences in oral tribology related research in industry and in academia? So in terms of maybe approach or priority and techniques, and also how can one guide the other? Thank you.
That's a very interesting question. So I can only speak from few of the industries with which I work with, and I know most of the industries work with Anton Power MCR 302, where they just attach a tribocell and work on it. Issue which I face is some of the times they use it for a more routine purpose rather than spending time on understanding. So, you're not changing the surfaces, you're just doing on steel contact. Whereas in academia, a lot of effort has been put into to modify surfaces to make it similar to what happens in the mouth. So even if we get a correlation in industrial setting, what do you do with that correlation, to be honest with you, even if suddenly you get a correlation? So that's the problem. However, on a better note, there are a lot of industry academic collaboration that happens, and most of the big industries are always collaborating on fundamental science with academia. So there is a lot of cross transfer of knowledge happening and will happen even more, considering there has been tremendous progress in this area in the last couple of years, and not only in tribology, but in elsewhere also.
Okay, thank you so much.
Thank you, Yug. And then we have one more question from Dr. Chatterjee. I hope I pronounced the name correctly. So you can unmute yourself and ask the question. Okay, if that's not going to happen, then I just read the question. So it says the following: very nice informative talk by Professor Sarkar. I want to know, is the contact stress uniform throughout in the time?
No.
And no. Oh, that's very short. That's very, very short.
Yeah, because that's why I gave the differences, because your filiform and fungiform, they are an order of magnitude difference.
Yeah, I can imagine. And another question is, what could be the possible lubrication regime during astringent conditions? Could you talk a little about it?
Let's put it that way. The hypothesis is it should be in boundary regime, but we don't know. There is very limited proof in this area.
Okay, then there will be another question from Marco. Marco, you can unmute yourself and ask the question.
Thank you very much for the inspiring talk, Professor Sarkar. And as regarding your conclusion, you were saying that you should build mechanistic hypotheses where we have, in order to have more information or not have misinterpretation of tribology sensory correlations. Could you explain a little bit more about that?
Sometimes in literature, there are relationships between even steel contact surfaces and sensory perception. And I am a bit of a person who feels like causation should be looked at than correlation. Because if you understand causation, you can build mechanism that why it is happening. If there is a correlation, that does not mean it will happen the next time, because the system might be different. But for example, if you think of hydration lubrication related to microgels, that should work for any microgel, irrespective of whatever microgel it is, provided you can define the boundary conditions. This is the deformability, this should be the size, and it should work. So for me, the main point is about, even if you get a lot of relationship in steel contacts and stuff like that, think it twice, because the system is far, far away from what is there in the mouth. That's the only point which I try to make from that comment.
Thank you very much.
Hi. So to follow up with that question, I would like to ask one more question. So there was this mention about that the tribology is always like a system property. So when I see the tests, what you have performed, which are mostly nano tribology tests, micro and nano level. Say, for example, if you're doing the same indentation test in the not in the nano or micro scale, like in the macro scale, how far are the properties varying? Do you think that, say, for example, if we are calculating the modulus values for a tongue-like surface, so the modulus values, when you calculate it in the nano or the micro, how far the values are varying?
Well, they're not. No, because they are material properties, so they don't change at all. So they are not scalable. Means they are scalable actually. So deformability is something, even if you do at nano indentation scale, they don't vary. And that's what we saw. What varies is the friction properties, because that is dependent on whether the relationship depends on the contact area or not. In most of the macro scale experiments, we see that they don't depend on the contact area at all. Whereas at nanoscale, since adhesion matters tremendously, then you start seeing all sorts of amazing things, which honestly speaking is very hard to understand as well without proper theoretical fitting. So yes, deformability doesn't matter. It's more like, of course, it will matter depending upon the system, but I'm more talking about the tongue versus the papillae. It doesn't. With PDMS, it doesn't. But friction properties, yes, it matters tremendously.
Thank you. I think there's one last question, maybe. They say, I want to know, it's from Chandrasekhar, and thank you, Professor, for such a nice talk. I want to know, is this possible to connect bulk rheology with tribology using some correlation?
So what we generally do, so in food, things are very, very shear thinning. They are not like fascinating Newtonian materials by any means. So what we do is we actually correlate with second plateau shear viscosity and try to correlate the relationship, particularly in the elastrohydrodynamic regime. But that's it. We have seen some relationship in the mixed regime, but that's it. Not any further. When you go to boundary, not much relationship we have seen.
Perfect. I think we are almost on time. So we did complete the Thank you, Professor. I mean, for me, myself being like a PhD, I mean, doing tribology related, and I'm also doing biotribology, and this talk was really very interesting for me. I hope it was also interesting for everyone who attended today, and thank you so much for accepting our invitation. This video, I mean, this recording will be uploaded on the TriboNet website, and we'll be sharing this link to everyone who have attended today. So thank you everyone for joining. And the next tribology related talk will be in the next month. It's on, I think it will be on the Earth tribology and earthquakes. I hope you guys will find that interesting as well. So please do join for that. And thank you all for joining once again. Thank you, Professor.
Thank you very much. Thank you. Bye-bye.
Thank you
See
very
you
much.
all. Bye.
Thank you, everybody. Bye-bye.
Bye. Thank you.